Cell adhesion is the ability or the process of the cells sticking either to other cells or to other structures. So in other words, adhesions can be cell to cell or cell to non-cell. When we say non-cells, we are referring to the interstitial matrix or the basal lamina of tissues. Based on functions, the cell adhesions or junctions can be classified as anchoring, occluding, channel forming, or signal relaying. Anchoring junctions are those that bind cells together and those that anchor the cell to the matrix. This includes adherence junction, hemidesmosomes, and desmosomes. Occluding junctions, on the other hand, are those that tighten the space between the cells. This includes tight junctions. Channel-forming junctions are those that allow cytoplasmic sharing between cells. These include gap junctions. Finally, signal-relaying cell adhesions are those exhibited by neurons or neuron connections. This is how a tight junction looks like up close. It serves to tighten the space between cells, which helps regulate passage of molecules between them. The more tight the junction is, the less molecules it allows to pass. Some are so tight that even water cannot pass through. This is the adherence junction or the zonula adherens. It anchors adjacent cells to one another through intercellular microfilaments. Desmosomes, on the other hand, are patches characterized by opposed thickenings of membranes of two adjacent cells. They anchor the cells to intermediate filaments. And these are hemidesmosomes. They bind cells to the matrix or the basal lamina of tissues. Finally, these are the gap junctions. These junctions allow cell-to-cell sharing of cytoplasmic content, as mentioned earlier. They have tube-like structures called connexons that are formed by 6 rings of connexin monomers. As you can see in the middle, there are holes wherein the cytoplasm can be shared between two cells. Cell adhesion molecules, or CAMs, are molecules that enable cell adhesions or junctions. CAMs in animals fall under 4 superfamilies of molecules. So we have the cadherins, immunoglobulins, integrins, and selectins. Cadherins and immunoglobulins are homophilic CAMs as they directly bind to the same type of CAM in another cell, while integrins and selectins are heterophilic as they bind to different types of CAMs. Defects in cell adhesions are usually attributable to failure in the expression of CAMs. Now let's go to cell movements and motor molecules. So cell movements, the most observable are in animal cells are muscle contraction and movement of the flagella and cilia. So these two are the basic types of motor molecules that are involved. These are the actin-based molecules and the microtubule-based molecules. The actin-based molecules includes the myosin superfamily, and they are almost all involved in muscle contraction. While the microtubule-based molecules includes kinensins and dyneins, and they are involved in the movement of cilia and flagella and movement of organelles. In this background, you can see a kinensin moving a vacuole along the microtubule. It's like walking on microtubule. So, That's it for now. Thank you very much for taking time to listen. Our next topic will be topic 5, cell processes.